Okay, I would like to thank the organizer and the session chair for giving me the opportunity to share the data with you. So I'm going to talk about one of our recent work on genome-wide analysis that we have identified a post-transcriptional operon as an endogenous defense against inflammation. So as Anton said in his previous talk, that expression of mRNA may not always faithfully reflect protein synthesis. So our genome-wide analysis, we have done a genome-wide analysis by polyribosome profiling, and we identified a regulation of a cohort of genes that is encoding inflammatory protein, and their regulation is very consistent with the signature that mRNA, expression of mRNA, may not always faithfully reflect protein synthesis. And the enormous implication of this regulation in number of disease, human disease caused by inflammation. So I would like to draw your attention to a very old data that we have, we got at least a decade before on translation regulation. So this is what you are seeing, human monocytic cells. That monocytes are very important cells of our innate immunity, that if you treat the cells with interferon gamma, you can induce ceruloplasmin mRNA. This is an inflammatory protein in monocytes, also made by liver. And however, you can see the mRNA is induced, but if you do a de novo protein synthesis analysis by pulse chase of the pulse chase study with S35-methionine of the newly synthesized protein, you can see that 8 hours after induction there is abundant message and the new protein synthesis is being made. However, even after 24 hours of induction, even there is abundant message but no new protein synthesis, showing that there is a translation control might exist. Since this talk is about translation control, I would like to just remind you about the 101 of translation, that translation is divided into 3 steps, translation initiation, elongation, and termination. So in the initiation step, starts with the formation of the EIF4F translation initiation complex that begins with binding of cap binding protein with the M7 GTP cap at the extreme 5' end, and that triggers the formation of EIF4F complex with the EIF4G, 4A, and the recruitment of the small subunit 40S bound EIF3. This is another translation initiation factor. Once this complex formed, this complex could scan the 5' untranslated region of a message, and when it comes to the start codon, GTP hydrolysis happen, and then all initiation factor falls off except the initiator tRNA bound to the 40S, and that allows the large subunit to join, 60S join, and 80S complex formation happen, and then elongation starts with the extension of protein chain according to and the cognate tRNA being added, and in the termination side there is no cognate tRNA exists, and the ribosome fall off. However, this process can be regulated at the level of translation by directly binding the many RNA binding proteins either at the 3' UTR or at the 5' UTR. So today I'm going to talk about one of our studies that involves binding of a RNA binding complex in the 3' UTR and how it could control the expression of a cohort of inflammatory gene at the level of translation. So what we have identified in terms of mechanism, that if you treat human monocytic cells with interferon gamma, it causes the release of a ribosomal protein L13A, which is the protein of large ribosomal subunit. This protein got phosphorylated and then joined with a multi-subunit complex with GluPro-RS, NSAP1 and GAPDH, and forms a multi-subunit RNA binding protein, and that binds the element, we name it GAIT element, gamma activated inhibitor of translation element. This element is staying in the 3' UTR of many target message. So whatever message harbors this 3' UTR will be a potential target of this regulatory mechanism. So I'm not going to in detail of this because this is an old story that we have published long time back. So just to take the long story short, what we have identified, the detailed molecular mechanism of this study, that we have identified that in the 3' UTR assembles this GAIT complex, which is a RNA binding protein complex, and with L13A is a significant component of that complex. This L13A, as a part of this complex, could bind the EIF4G, which is the translation initiation complex, and it occupies a position where it is the position of joining the 43S subunit complex. So by virtue of binding to EIF4G, this complex inhibits the recruitment of 43S subunit, thereby inhibits translation initiation of all the message that harbors the GAIT element here. So such a sophisticated mechanism may not have evolved only to silence one gene. So we hypothesize there could be a genome-wide target of this mechanism might exist. And we have undertake a very, very unbiased approach to identify other targets. So this is by polyribosome profiling. So this is, we know that a translationally active mRNA got to be associated with the polyribosome in order to synthesize protein. So the polyribosome association of a particular message is the quantitative measure of the translatability of that message. So by measuring the ratio of a particular message, ratio of polyribosome versus non-polyribosome association could give you a quantitative measure of how efficiently that message could be translated. So we did exactly that here. So we treat human monocytes or macrophage with interferon gamma, treated or untreated, and then resolve the cell by a translationally active polyribosome or inactive, translationally inactive silence pool. These are the different ribosomal subunit. This is the polysome. So we isolated all the mRNAs associated with the polyribosome. That means all of the mRNA that is actively translating under this condition. And we measured the ratio. So what I will show you, the ultimate data of the bioinformatic analysis that shows. So this is a data of the Affymetrix Gene Chip analysis after the polyribosomal and non-polyribosomal mRNA. So here what you are seeing is the unfractionated mRNA upon interferon gamma treatment. So this scale is green is less and red is typical heat plot scale. So you can see that all of these genes are induced in response to interferon gamma, just if you are considering unfractionated RNA. But if you are considering the ratio of polysomal versus non-polysomal mRNA, which is actually the quantitative measure of the translability of particular mRNA, you will see even they are induced, but they are not actively translating. So at 18 hours upon interferon gamma treatment, showing that all of this message could be a target of translation regulation. So we have validated this using real-time PCR approach, that many of them are inflammatory genes. See, these are the chemokines and the chemokine receptors. So they are induced in response to interferon gamma, but if you are looking at their polysomal abundance, they are less at 18 hours after interferon gamma treatment. So showing that these could be the potential target of the translation control mechanism. So however, we know that this control mechanism is driven by the release of L13A. Are they L13A dependent? So in order to ask that question, so we did again a high throughput analysis using inflammation array analysis, using the cells that do not make L13A. Here we have knocked down the monocytes with by using shRNA approach, by RNAi approach. Here the cells do not produce L13A, and then we have done the same analysis, and we are measuring the polysome non-polysome ratio, where you are seeing here in control cells their ratio is very less, showing they are not efficiently translated. But when you remove L13A, so all of a sudden the ratio is high, showing this L13A is playing a role in blocking the protein synthesis of all these inflammatory genes. So in order to ask whether this mechanism is important in physiological system, so we have generated a macrophage-specific L13A knockout mouse using the CRE-LOX system. I'm not going into that detail, but what I'm showing here, the ultimate data that this is the macrophage of the macrophage specific L13 knockout mouse, and this is the liver and kidney of the same animal. And we are using immunoblot analysis that you can see here that the L13 is not present in the macrophage specific knockout mouse. However, in liver and kidney, the expression of L13 is uninterrupted, showing that we have successfully created a tissue specific knockout mouse of this protein. And this is the design how we have done it. We actually knock down the entire, we put the LOXP site between the exon 1 and 2 and after exon 8, and as a result when we cross this with CRE, because the CRE recombinase here is driven by the lysogen M specific promoter, which is a myeloid cell specific promoter. So it will not, CRE will not express in all other cells, but will be only expressed in myeloid cell. We have tested all of this. And this is actually the final result of this mouse. So now I'm going to show you a couple of data from this mouse. So this mouse is viable, breeding competent, and indistinguishable from the control mouse under the normal or unchallenged condition. If you don't do anything, both of the mouse are growing well, they are perfectly breeding competent, and we couldn't see any difference. But when we challenge this mouse with endotoxin or LPS, then the scenario is totally different. So now I'm going to show you a couple of experiments showing that how we subjected this mouse with lipopolysaccharide treatment and induced systemic inflammation. Because why? Because LPS is also a potent inducer of interferon gamma. So this is the first analysis of this mouse. We have done a Kaplan-Muir survival analysis of this mouse that shows that this knockout mouse is showing significantly decreased survival rate upon LPS challenge. So these are some of the histopathological analysis of the different organs of the mouse. Here you are seeing the HN stainings of the blood vessel of the lung, that you can see that there is huge blood clot we saw. And also this is the bronchus. You can see that in the control mouse, the bronchus is more open and most of the RBCs are intravascular. But in the knockout mouse, the RBCs are most of them are outside, showing bleeding. And also we can see that when we stain this with MIP2, which is the neutrophil specific marker, that you can see that the neutrophil invasion inside the blood vessel. And this is a staining for macrophage by MAC2 antibody, and this is a blood lining of the blood vessel of the lung, that you can see that inside the lung vessel, the macrophage invasion of macrophage, showing that these are under highly inflamed condition, these mouse. If you challenge them with inflammatory stimuli, with a sublethal dose, in the control mouse, you don't see that much pathology, but in the knockout mouse, showing very high pathology. So we looked at the kidney glomeruli. Here we can see in individual kidney cells, kidney glomeruli cells, and you can see here this is the expanded version of this, that you can see that high magnification, that you can see that very significant infiltration of macrophage inside the kidney glomeruli. And also, this is consistent, with other data that shows, severe kidney failure in this mouse. So infiltration of neutrophils and macrophage in the tissue peritoneum are also the hallmark of inflammation. So what we have done here, we asked whether induction of systemic inflammation or septic shock by LPS, what happens to these immune cells in the tissue. So we have collected the peritoneal immune cells, total leukocytes, and then do FACS analysis using GR1 CD11B or a 480 GR1 or a 480 CD11B marker specific antibody. And you can see here that significant increase of the double positive cells, showing that these mouse are under highly inflammatory condition. So, but are these due to the deregulation of the GAIT target mRNA? So here we have collected induce the mouse with LPS and then harvest peritoneal macrophage after 24 hours and started the culture immediately and collected the conditioned media from the plate. So showing the steady state level of these chemokines. And we have measured that the knockout mouse shows significantly increase steady state level. However, this experiment do not directly show that they are under translation control, because steady state level only reflects, the amount at that moment. So, but to identify translation control or to test whether this mouse is also under translation, showing translation control of these chemokine and chemokine receptor, we did the ultimate experiment, that harvesting the macrophage from this mouse and then do a polysome analysis of the primary macrophage of the control mouse and the knockout mouse, and then collected fractions, and then do the RT-PCR analysis using 3 known GAIT target mRNAs, CCL2, CEX-CL13, CCR3. What you can see here in the control mouse, most of the messenger RNA, they are induced, but they are not actively translated, because most of them are associated with the free fraction, means translationally inactive fraction. But in the knockout mouse, there is no L13A in this macrophage, in this mouse. And you can see both of them, all of them have a tendency to accumulate into the translationally active polyribosome fraction, showing that this mouse lost their ability to control at the translation level of these chemokine and chemokine receptor. As a result, they are showing increased, significantly high steady state level and other downstream effect of pathology, inflammation related pathology. Now I'm going to show you just one more example. You know that these days the new thought is the cardiovascular disease, particularly atherosclerosis, is also an inflammatory disease. So this is coming from the high inflammatory state of the cells that is residing in the blood vessel wall. So we monitored the atherosclerotic plaque formation of this mouse. So what happened if we chase this mouse in induced atherosclerosis by feeding this mouse with high-fat diet? So in order to do that, we first breed this mouse in ApoE background, because ApoE, you cannot induce atherosclerosis in mouse until and unless you breed them in ApoE knockout background. So this is the hallmark studies from many labs. So we breed this mouse in ApoE background, both of them, and then feed them high-fat diet, collect the heart, and then here we have conducted the aortic root analysis, where you can see the heart valve, and here you can see the deposition of the fat of the atherosclerotic plaque. So we have identified significantly higher amount of the deposition of atherosclerotic plaque in the heart aortic root of this mouse. So this is the quantification of the data. And not only that, we have done an NFAS analysis, where you are now monitoring the entire aortic tree that is going from the heart to all the organs. So you can see significant blockage of this. So human also have that at the time of before heart disease, that this ultimately block the blood flow and can cause myocardial infarction. So this is showing that presence of L13A in the macrophage could act as an endogenous defense mechanism against all of these inflammatory disease. So same thing, we have done the translation status of this target mRNA in the high-fat diet feed mouse, that the knockout mouse shows consistently higher abundance of this in the polyribosome fraction, showing their translation status is high when L13A is not there. So I will conclude my talk with this model that showing you that inflammation is also important. This is the important part of our host defense, that inflammation causes the activation of many lethal molecules, and we need that to fight the invading organism. However, if it is uncontrolled, it could kill the host. It could do harm to the host. So what we believe here, that we could induce this member of the post-transcriptional operon at early hours after inflammatory stimuli, and this is important for the host. However, in the resolution phase, this GAIT complex that which L13A plays a critical role, turned on at late time point, and then it can bind the 3'. All of these message hovers this GAIT element, and it could bind the 3' UTR and shut down their translation simultaneously. So this is a very unique mechanism that by triggering translation control of GAIT complex mediated translation control, cell could block the synthesis of a cohort of inflammatory genes simultaneously. So I'm not going to show you one of another data. So this is a very recent work that we have also found that in human colitis, that we induce colitis in the mouse, and we have seen the importance of this. But that data, I didn't have time to show that data. So with that, I'll stop here and I'll acknowledge my lab. Darshana Poddar, the graduate student, and many of them have contributed in this work, and I'm also grateful for the National Heart, Lung and Blood Institute for R01 grant and American Heart Association for their Scientist Development Grant and Granting Aid, and several of my pre-doctoral and postdoctoral fellows are also awarded from American Heart during the training in my lab. And thank you. I'll entertain any questions. Thanks.
Are there any questions?
To the humans about for your work from mouse model to the humans, is it how easy?
Yeah, we of course envision that, but at this moment, our goal is to identify. So we have a reporter-based assay system to identify the translation regulation, that we have a luciferase fused to the 3' UTR. So we are very much interested to screen small molecule library which can induce translation control, and once we have that, we'll test this in our model mouse system, and then that's my dream, that one day it will go to the clinical trial.
Any other questions? Yes, please.
So how many genes do you think does L13A, the ribosomal protein, regulate? How many genes does it regulate, do you think? And is it only 2 inflammatory genes, or does it have any other function besides it?
See, I didn't show all the data, but the analysis, the bioinformatic analysis of the genome-wide study, at this moment we are focusing only on the inflammatory gene, because most of the gene, this is not the only list. We have also several more, maybe 40, 50 genes list we have, but what we are showing here that we could validate in animal model, but we have more list that is regulated by this, but we don't know the function of those genes.
But considering that this is a ribosomal protein, I think that it regulates almost every gene, but
no.
Do you think like that?
No. So this is a very important point. So when this ribosomal protein, although is a part of the protein synthesis machinery, but it is not taking part into the generic protein synthesis. So I didn't share those data. We have made knockdown cells, that L13 a knockdown cell, and monitor protein synthesis, generic protein synthesis. It doesn't screw any of them. Protein synthesis, generic protein synthesis remains same. So that's what our hypothesis is. So these kind of ribosomal protein, their role in protein synthesis is dispensable, but that's why their other extra ribosomal role have evolved for this protein. So but this is a very good question, and but L13A do not have any function in generic protein synthesis. So that's the answer I can say now. Yes.
Respiratory disease models like OVA-induced or
besides
the LPS.
We have not tried any respiratory disease model, but we recently published another paper in Journal of Virology that shows that this mechanism is important for a important respiratory virus called respiratory syncytial virus, and also human para-influenza virus. So we are pursuing that stuff too, and collaboration with the virologist.
So I also want to know, have you done any lung function test?
Lung, we have not done any lung function test, but the lung of this mouse is very weak with little bit of inflammatory stimuli. But under normal condition, it's good, you don't see any difference between them. But when we challenge them, infection model, apply infection model to them, then you see it's totally different scenario. Their organs, lung, kidney, all of these major organs are severely affected in this mouse.
Just a curiosity, do you find any heterozygote, or have you done those type of experiments?
Yeah, we recently have, in the process of making total knockout mouse, we just identified the heterozygous, and we are trying to breed them in the homozygous model. But we searched the SNP database, and we didn't find any SNPs of this L13. But that may not necessarily mean that it is not exist. It only shows that it's not shown in the database. Okay.
Any other questions? Okay, thank you.
Thank you.